This is our second class on molecular basis of inheritance. In our previous class, we have discussed about the experiments in search of genetic materials. That means we have discussed the experiments of Griffith and Avery. Now we know that Avery concluded that the DNA is the transforming principle. But that time, most of the scientists believed that the protein was the transforming principle. So, in 1952, Hershey and Chase, two scientists, designed an experiment that provide more evidence that the DNA is the genetic material. Before starting their experiment, we need to know some basic things about viruses because they used T2 phage. What is bacteriophage? What is phage virus? Phages are the viruses that infect bacteria, and they have very simple structure. Suppose this is a bacteriophage and this is their protein coat and this is the genetic material. It could be DNA or RNA. This is bacteriophage that infect the bacteria. Virus reproduces through lytic and lysogenic cycle. These T2 virus reproduce through lytic cycle. We will make a complete video on lytic and lysogenic cycle. You can check it. But for this experiment of Hershey and Chase, we need to know the lytic cycle because T2 phage reproduce through lytic cycle. Suppose this is a bacteria, E. coli, and this is a bacteriophage that infect the E. coli. E. coli has its own chromosome, DNA, circular DNA, and when these phage infect the E. coli, it just inject its DNA or genetic material. It will produce some protein that actually destroy the bacterial chromosome. Now this phage genetic material will reproduce and makes many copies, and then it produces protein that will make more bacteriophages, and then it will destroy the bacterial cell and get released. This is the process they reproduce. Now we will know the experiment of Hershey and Chase. In their experiment, Hershey and Chase used T2 phage. They knew that the phage has only protein and DNA, and when they infect a bacteria, they just inject their genetic material, that means the DNA, into the bacterial cell. As we know that the protein may have sulfur but not phosphorus, and DNA has phosphorus but not sulfur. So Hershey and Chase used their isotopes, that is As35 radioisotope of sulfur and radioisotope of phosphorus, that is P32, and they designed two batches in their experiment. They labeled T2 phage with the radioisotope. So suppose this is the bacteriophage, which is labeled with H35, and this is the bacteriophage which is labeled with P32. Now they start the experiment. This experiment has 3 steps. Number 1 is infection. In this step, the bacteria is infected by bacteriophage. In both cases. So they leave them for some time to reproduce. So many copies of bacteriophages are formed, and now in the next step is agitation. In this step, they blended the solution to detach the bacteriophages from the surface of bacterium. So now this is the bacteria with many bacteriophages inside them, and no bacteriophages is attached to their surface. The next step is centrifugation. What is centrifugation? Centrifugation is a process in which centrifuge, which is a machine, is used. In this process, centrifugal force is used to separate particles according to their shape, size, viscosity, etc. So when they centrifuge it, the heavier bacteria of this solution will be settled down in both cases, and lighter bacteriophages will be suspended in the supernatant solution. This black dots is bacteriophages. This is the experiment. Now this is the time for observation. They found no radioactivity here. But they found radioactivity of P32 here in this batch. So it is clear that H35 is not transferred from the phages to the bacteria, and P32 is transferred from the phages to the bacteria. That means DNA is the genetic material. As we know, the P is only present in DNA. So the DNA is entered the cell but not the protein. So they concluded that DNA is only the genetic material. So tell me, why did Hershey and Chase particularly use the radioisotope of sulfur and phosphorus? Comment your answer. Now it is clear that the DNA is the genetic material. But why DNA? Why not RNA or proteins or other molecules? That means there are some criteria for a molecule to be genetic material. What are the criteria? Number 1, ability to replicate. Number 2, structural stability. Number 3, ability to mutate. And number 4, ability to express their proteins. Now we will see which one is better, DNA or RNA. Or RNA. The first criteria is to replicate, ability to replicate. What is the meaning of replication? It means to make exact their own copies. DNA and RNA both can do this. That means both can replicate. The next criteria is structural stability. As we know that RNA has OH group in their 2' of pentose sugar. This OH group is unstable, while DNA has H, only H group in this 2' position. So DNA is stable, and again RNA has uracil, while DNA has thymine. Uracil is more unstable than thymine. So for this structural stability, DNA is better genetic material than RNA because it has H group and thymine which is stable. The third criteria is to mutate. Both DNA and RNA can mutate, but as RNA is more unstable, so it can mutate easily. Those who have RNA as their genetic material, like virus, for example, corona or HIV, they can mutate easily for this reason. Number 4 criteria is to express proteins, the ability to express protein. Both can express protein, DNA and RNA, but DNA has to synthesize RNA at first, that is mRNA, and then this mRNA will produce protein. But for RNA can synthesize proteins. For DNA, it has to depend on RNA. So from this chart, we see that both can replicate, both can mutate, both can express. So both can be genetic material. But as DNA is more stable than RNA, so we can say that DNA is better genetic material. Now the question is, which was the first genetic material, DNA or RNA? RNA used to act as a genetic material as well as a catalyst. As we know, enzymes are the biocatalyst, ribozymes are the RNA that act as an enzyme. During evolution, DNA evolved from RNA with chemical modification, and that's why DNA is more stable than RNA. So RNA was the first genetic material, and this hypothesis is called RNA world. Now the question is, why protein is not considered as genetic material? Comment your answer. In our next class, we will discuss how DNA replicates. To understand the entire chapter, you have to revise your knowledge of biomolecules. Thank you.